So I'm just going to begin by doing an acknowledgement of country. So Melbourne Water acknowledges the Victorian traditional owners and their elders, past and present, as the original custodians of Victoria's lands and waters. And I pay my respects to their elders, past and present, and to the ongoing living culture of Aboriginal and Torres Strait Islander people. And I'd just like to welcome any Aboriginal or Torres Strait Islander people we have with us today. So welcome and thanks for coming. So thank you for joining us today. So we're going to hear from Frank Amtstaetter, and he's going to talk about the fascinating life history strategies of some of Victoria's native fish species and the effect of flow on migration. So Frank is a senior research scientist at the Arthur Rylah Institute for Environmental Research in Heidelberg, where he's worked for 12 years. His focus is investigating the effectiveness of environmental flows and barrier remediation in promoting our native migratory fish species in coastal rivers. So prior to this role, he worked in Ontario, Canada for 13 years, investigating the effects of various stressors on fish populations. So I'd just like to give a big welcome to Frank, and yeah, I'll hand over to you. Thank you.
Great. Thanks, Teresa. And thanks everyone out there who came to the webinar today. As Teresa mentioned, I'm going to be chatting about the life history strategy of some of our species that live in coastal rivers. And the ones that I'm speaking about in particular today, they need to migrate between fresh water and the ocean. So having that migration pathway open is really important for these fish. And then I'll be touching on a couple of the potential threats to these species in barriers to that migration and modified flow regimes. So we'll start off with a bit of a background on the life history strategy of these fish, and then I'll get into some case studies, some of the work that we've done in the last 5 to 10 years, investigating how barriers can impact these fish and how we can remediate that, and how modified flow regimes impact these fish. And in these case studies, it'll give you a good idea how we go around collecting information and then how that information can be used in the management of these fish species. So we'll start out with a bit of background information. So these fish species that migrate between freshwater and the ocean, they're called diadromous fish, and quite a high proportion of our native fish in our coastal streams are diadromous. So, maintaining that migration or those migration pathways is really important for protecting a large proportion of our fish in coastal rivers. So we have a wide variety of diadromous fish, these fish that migrate, but I'm going to focus today on a group that they reside in fresh water for most of their lives. So we have other species that reside in the ocean for most of their lives, but I'll just focus on this one group today. And although even within this one group, their habits vary, most of the fish, they migrate downstream to spawn. And then that spawning can occur in these sort of lower freshwater reaches. And I'm going to be using my mouse today to try to point some things out in the slides. Some of them will actually spawn in the estuaries, and some actually, they migrate right out to sea and continue their migration before they spawn. But the important bit is that larvae of these fish all end up in the ocean. And then these fish will spend, well, the time varies, but at least several months in the ocean. And then as really small juveniles, they need to find these rivers to migrate or immigrate into them and then disperse upstream. So we have these three very important processes: the downstream spawning migrations, the attraction and immigration into streams, and then this upstream dispersal that allows them to move throughout the rivers. And you can imagine that if any of these processes are interrupted, it can really hamper the abundance of fish and their distribution within our coastal rivers. So this image is of Dights Falls, where there's a dam. So you can imagine if you're one of these juvenile fish that are migrating into a river, and you make it a couple kilometers into the river, and you just come across this concrete wall, and that'll completely block you from reaching hundreds of kilometers upstream of that. So you can imagine, as a result, you won't get fish upstream of that structure. And on top of that, downstream in this area, you still have all these fish moving into the river. And if they can't get any farther, you just get loads and loads of fish being literally trapped in this area. So then that increases the mortality of these fish. You can imagine that birds can come in and start feeding on them. And even aside from predation, there's competition for resources. So as a result of these high densities, you have much lower survival. And then the other thing that we're going to talk about are modified flow regimes. So in this case, you can have a dam much higher in the watershed, and you can imagine that, say, in the springtime or late winter, when we get most of our rainfall down here, rather than that water flowing out into the ocean, it's being collected. So when these fish are trying to find a river to swim into, and maybe you have 10% of the water volume flowing out, it can be harder for them to pick up that chemical signal and actually find these rivers that they want to immigrate into. So those are the two threats that we'll be talking about a bit today. And then the next step will be to get into some of these threats, the barriers, and modified flow regimes, and some of the work that we've done, some of the research that we've done. So Tim, I reckon if anyone has a question, this could be a good time to take a short break and talk about some of the life history strategy things around these fishes. Okay, well, I couldn't hear anything, Tim, so I'm going to assume that we don't have any questions to deal with at the moment. So we'll start talking about barriers first. And so we know that barriers can block the movement, the migration of these fish. And there's some things we can do to help fish get over or around these barriers. And one of the big ones, for large barriers anyway, are fishways. Now, there are a couple types of fishways. The two images at the top, that's the installation of a vertical slot fishway, as well as the one in the bottom left. And these fishways provide a pathway for fish to get around the barrier. So these ones, they're made out of concrete, and you can see where my mouse is, they're made up of several pools. And if you could hopefully just see that gap, so that's a slot connecting these two pools together. So these fishways are designed to limit the water velocity moving through that slot. So even our really small juvenile fish that are migrating upstream can swim through it. And they're designed so the turbulence within each of these pools is low enough so the fish can get in, have a rest, swim up into the next pool, and that way they can move up through each of these series of pools and out the top end. Now, with larger barriers or higher barriers, you need more of these pools. So if this example on the bottom left, the entrances are on this side of the fishway. This was before it was fully commissioned, and they've got to swim through a whole series of these pools out the back and back up to the top to the exit of the fishway. So, these fishways can pass fish by barriers that are several meters high. It just takes more of these pools. And like I said, before I say that, then on the bottom right, this image is of the installation of a rock ramp fishway. And they differ from the vertical slot fishways in that basically the river below the barrier is filled in. So rather than having, say, a 1 or 2 meter wall, that 1 or 2 meters is spread out over, it could be 100 meters by filling with rock. And that provides channels that the fish can actually swim through, and then they can swim right up to and over top of the barrier. In addition to these really large barriers, we also have almost countless number of smaller barriers and things like culverts in tributaries of some of our bigger rivers. And you can imagine, a culvert like the one in the photograph. These have very smooth sides, and it actually funnels the water in the stream into the culvert, and that results in, or can result in, really high water velocities. And then these really small fish that are trying to migrate upstream, they just can't swim that quickly for that long a distance, and it blocks their movement upstream. So rather than using fishways for this, there are some other designs that have been used. And this is actually in Cardinia Creek, just east of Melbourne. And what they did for this one, they actually built a rock ramp fishway, which is the photograph on the bottom right, downstream, just downstream of the culvert. And as you can see, there are these ridges of rocks. So you have the pools in between. So fish can swim between these rocks into these pools and get up the top end. But by installing this fishway, it backed up a fair bit of water. And that resulted in the water in the culvert being deeper and having less flow, because it's backed up. So it's more like a slow flowing pool than a raging torrent through the culvert. Now, the other thing they had to do, because there were still some flows at the top end of the culvert, and this photograph on the bottom left shows this, they installed a series of baffles. So since there was still a little bit of flow at the top, they put in these baffles to break up that flow so fish can swim up and then swim around these. And if they need a rest, they can hide in behind one of these baffles, and then they can swim out the top. So that's a way to get fish swimming through some of these smaller structures that can block their movement or their migration upstream. So it's great that we have these techniques to help fish get around or through these structures, but we really need to know whether they work or not. And that's some of the work that we do at the Arthur Rylah Institute, because we spend a lot of money and a lot of time is spent to fix some of these issues, but we need to know whether they work. So we know whether we should continue doing these kinds of works, or if they don't work, how we can modify them. So for fishways, there are several things that we can do to determine whether they work. And one of them is we can catch fish moving through the fishways. So we can use traps in the cells of the fishway to catch fish that are moving in. And it involves trapping various cells of the fishway. So we can trap the entrance, or the very bottom end of the fishway, partway through, and the top end. So what we're looking for is to see if the number of fish entering the fishway is the same as the number of fish exiting at the top end. And so some of the things we found, and this is in a couple of the examples I showed earlier, those two vertical slot fishways in a previous slide. When the migration of these juvenile fish is really happening, we can catch thousands of fish per hour moving in and through these fishways. So it just gives you an idea of how many fish are moving into and up these rivers. In both these cases, we didn't find differences in the catch rates of the fish entering the fishway and those swimming out at the top end. So that tells us that the fishway as designed is working really well. So fish that get in can swim all the way through. It means that there isn't too much turbulence in those pools. It means that the water velocity moving through the slots is within their swimming ability. So that's great. And we also catch a really wide range of sizes. So fish from 20 millimeters, which is smaller than most of the fish we're talking about today, those juvenile migratory fish, up to the eels that are up to almost half a meter in length that we've caught moving through these fishways. So like I say, it tells us whether these fishways can pass fish and whether they're designed well and built well, but it doesn't give us much knowledge around the passage efficiency overall. So are there fish that can't find the entrance? And so we've had some indications in the past where it appears that during really high flow events, fish can't find the entrance. And if you get indications like that, then you're going to have to determine why and try to fix things. So the other thing we can do, aside from determining whether fishways can pass fish, is look at relative passage efficiency. And this is some work we did in at that culvert site that I showed you earlier. And the photograph you see here on the right, or the only photograph, that's the top end of the culvert. And so we have this net that we had custom built, and it's got an aluminium frame, and that goes flat across the concrete, the concrete apron of the top end of the culvert, and it slots into some U-channel that was bolted into the apron. So fish that come up through the culvert had to swim in this net. So what we did for this one, it's a bit more involved than just doing catch rates at various places in a fishway. It involves catching some test fish downstream of our sites. We actually mark them with dyes, and we had to do that so we could determine if fish we caught subsequently were our test fish. And then we released them at our barrier site where the culvert is, and at a control site. And the reason we need control sites is so we have basically a baseline to compare with. So if we see differences at the test site, at the culvert site, we know whether it's just because it's not because it was just a better year for migration. It has to do with actually the fixing of the remediation of the culvert. And so we did this work before the work was done at the culvert, which was that installation of the rock ramp fishway and the baffles being installed, and then again afterward to see if we could see a difference. We had to do this a number of times. So each trial, we released 50 migrating galaxids. So these are those juvenile fish that are dispersing upstream. We released them at the bottom end of these sites, and the sites were about 75 meters long. And then we caught them at the top end of the sites over a 24-hour period. And we did that 5 times at each site, both before and after the work done at the culvert. So the photograph you see on this one, this is actually the recapture net at our control site. And we had to be very careful, similar to that culvert site, that we caught all the fish moving up through this section of the stream. So the bottom of the wings, they had some really heavy galvanized chains sewn into them, so it stayed flat on bottom. And then the wings were set tight to the bank. And in some cases, when the water level came up, we actually had to pack in behind the edges of these wings with netting material to make sure fish couldn't swim around. So it was really important that we caught every fish that moved through, so we could measure the movement efficiency or migration efficiency through these sections. So what we found out with this project was that the passage efficiency at the culvert, it increased quite dramatically. So before they did the work at the culvert, on average 3% of our fish were able to make it through the culvert. After the works were completed, 41% of our fish managed to swim through the culvert. So that's a huge difference. And in addition, that 41% is very similar to what we observed at the control site. So a site where just a natural part of the stream. So that indicates that remediation at the culvert has made this section of stream passable, similar to other natural sections of the stream. So that was a really good result. And the other thing we noticed while we were doing this work was at our culvert site, if we had a rain event at times, our passage efficiency appeared to be higher during those small increases in flows. Now, we didn't do enough sampling to be able to test that using this technique, but it got us thinking about how we might be able to test for that in the future. And when we get into the next section, which is about river flows, I'll be going over an experiment that we did to try to determine how effective increases in flow pulses can be in getting fish to swim upstream. So these first 2 examples really speak to how well fishways work, or remediation works, in passing fish around or through barriers. But that doesn't answer one of the big questions, and that is, so what? Fish get through. We expect that'll have an impact on the populations, but does it? And so that's what this third technique can do, and it involves sampling populations to determine abundance of fish in the rivers and look at their size structure. And this is upstream of where culvert or fishways have been installed, culvert remediation or fishways. And in this case, it was that big vertical slot fishway at Dights Falls. And this approach takes a number of years of sampling, so you can get an idea of what the population or how the population is changing. So we go in there and we have several sites upstream of the fishway, and we have to once again do it before the fishway is installed and then for several years after. But we also have control sites in areas that are unimpacted. So we go in and sample fish, and then we identify, we measure, and we release again at site of capture. And I'll just show you a map. This is of the Yarra River and its tributaries where this investigation took place. This red arrow shows you where Dights Falls is, where that vertical slot fishway was installed. And as I said, we have 4 sites that are unimpacted by it, and those are our control sites. And then we have sites within tributaries. So that we had 4 sites in Mary Creek, 4 sites in Diamond Creek, Mullum Creek, and then 4 around in the Warrandyte area, and then also up here at Stringy Bark. So our sites were up to 90 kilometers upstream of Dights Falls where the fishway was installed. So some of the results we found, and I'll start by talking about fish abundance. Now, this graph that you can see here, this is a graph, or these are results for our control sites, so the ones that are unimpacted by the new fishway. So this left axis here, that's catch per unit effort. It's really a measure of how many fish are at our sites, the number of fish per hour of sampling. This vertical dashed line indicates when the fishway was commissioned. And then I'd like you to focus on these solid, larger black dots. Those are our mean catches at those sites. And if you're interested, the smaller open circles just indicate the catch rates at each of our sites to give you an idea of the variation in the data. So as you can see, the first couple of years, one pre and one post, catch rates were relatively low, and they were a little bit higher over the last 3 years of sampling. So, and that could be just because there were more fish that were migrating into the rivers those years. But what it means is that if we're going to detect any difference as a result of the fishway, we would need to see a larger increase at our test sites, the ones upstream at the fishway, than we see in this control site. So the next slide I'm going to put up, it continues, it includes the results from our control sites, but it also has the results from the other 5 tributaries or locations in the Yarra River. So as you can see, in Diamond Creek, Mary Creek, and in all these other systems, the catch rates increased quite substantially by our last year of sampling in 2017. And these results were statistically significant. So that demonstrates that we've got more fish, more diadromous fish in the Yarra River upstream of Dights Falls as a result of the installation of the fishways. Now, the other thing we looked at was the size distribution. So these are photographs of common galaxids. And now that we have the fishways in place and working, we have, not only do we have more fish of all size classes, but the proportion of smaller fish in the population is greater than it was beforehand. And that's probably because, you know, these fish, they have quite a long duration for their migration. So they can start in August and go right through summer. So, and if they can continue migrating in or past the barrier for that entire period, all those months, you just build up more and more of these small fish that increase the size of the population and the proportion of small fish in the population. And in terms of healthy populations, you expect to see more of these really small fish, and then their numbers drop off as they get larger and get older. So these results have been very promising in terms of how fishways can improve populations at a much larger scale. So we're talking about reaches of river and tributaries. So once again, that's what where we're gonna finish with the work we've done on barriers. So if anyone has any questions before we get into the flows portion of it, we can take a small break and deal with some questions now.
Yep, we've got quite a few questions that have come through for that section. So the first one that we have is, what is the signal that juveniles are using to locate their point of entry for their return upstream?
Well, that's an excellent question, and there's a reason I skipped over it. We really don't know what that signal is, so we suspect it has to do with it's a chemical signal, and whether it's fresh water, so lack of salt, or whether it's a signal from conspecific, so fish of the same species, whether it's chemicals from those that they smell and say, okay, yes, I'm going to go up here because there's similar species up this river. We don't know that yet.
Cool. Did the design of fishways, was that tried in a laboratory scale before they were put into practice?
Oh, now the design of fishways, it's really been a long exercise, a little bit of trial and error. So early on, a lot of the fishway designs came from Europe and North America, and they were based largely on salmonids. So they didn't work well for our small fish. I don't know how much was done in labs versus how much trial and error was done in the creation of fishways. But I know some of the early ones didn't work well for our native fish because the water velocities were just too high. And our fish aren't jumping fish. So it really limited the success of those early fishways.
Cool. Has anyone tried or developed a mobile PIT system that can be placed at entry and exits of culverts so that pit tagging can be used for tracking the movement efficiency?
Yes, yeah, that can be done. There are a lot of, not only at culverts, but even at our vertical slot fishways. Some of the ones that we have installed actually have readers in them, so antennas, swim-through antennas and readers in the concrete. So if you mark enough fish downstream, you can pick them up moving through the fishways. I guess the downside in terms of passage efficiency, depending on the species, you don't know whether those fish are actively migrating upstream at that time or not. And for our juvenile fish, they're just too small to put pit tags into them.
Cool. A couple of questions around if there's the potential for environmental DNA collection to help with the sampling for some of these projects.
Yeah, so there could be, and that's you could use environmental DNA. It doesn't work well to detect or to determine abundance of fish. But if you're looking for some of the rare species, it's a way to determine presence absence. So if you can take enough water samples, then you should be able to pick up even the rare fish. And so the movement stuff, the population level stuff I spoke of, we were focusing on common galaxids. There are a lot of them out there, and for that kind of technique to work, you need to be able to catch quite a number of fish. But for some of our rare species like Australian grayling, that doesn't work. So the detection of those could be through eDNA or other sampling techniques during other projects.
Cool. So a lot of the stuff you were talking about with this project was the significant increase in the fish in the Yarra system past the fishway at Dights Falls.
Yep.
Have there been similar projects that you've worked on or that you know of in the Maribyrnong River?
No, there haven't. And looking at that population scale response, it's quite rare. It's happened in a couple of places in the States, but it doesn't happen. It doesn't occur very much at all. And part of that, I think, is because it does take quite a long time to collect the data.
Cool. Do you have data on the abundance and population structures of some of these fish species before the construction of these barriers?
Yeah, so for the Dights Falls one, as you would have noticed from the graphs, we only had 1 year of pre-treatment sampling, and ideally there would have been more. But by the time the fishway was contracted, we only had so much time for monitoring. Now there are other projects that occur in the Yarra River and other rivers. And prior to the installation of the fishway at Dights Falls, the capture of other diadromous fish like Australian grayling and tupong were incredibly rare. And even though we didn't pick up many, we picked up some, but we didn't pick up many in this project. But some of the electrofishing surveys done from boats in the main stem of the river, they've been picking up a lot more grayling and tupong that they'd seen previously. And when I say previously, we're talking over 100 years.
Cool, thank you. I've got a question about Galaxius in particular, and how far upstream do they need to go to be able to breed? And I'm guessing this is from Elizabeth, asking about some fish species in the Ulster Creek system. Probably most likely common Galaxius is the question that could be related to.
Yeah, so most likely common galaxids. And in terms of how far upstream, they don't have to go very far upstream. But the importance for that upstream dispersal is to increase survival. So some of the fish, they'll reside in the lower freshwater reaches, which is absolutely fine, and others will migrate some distance upstream, but it spreads the population out. So it really, the distance they move upstream doesn't impact their spawning. But when they spawn, they do migrate down toward the estuary, the top end of the estuary, to spawn. But they can do that from any part of the river that they get to.
Excellent. Yeah. So the question was around if the lack of a fish ladder in the Ulster Canal leading to the Ulsterwick Park could stop the Galaxius from being able to breed in the lake.
Right. So if they make it upstream of the barrier, could that trap them? Yeah. So these barriers can impact fish moving downstream. It doesn't happen as much, or it's not as critical as the upstream movement, because the upstream movement can just be stopped dead in its tracks. Where fish, if there's water spilling over top of the barriers, they might delay their migration, but fish can tumble over the tops of the barriers, which it's not ideal, but some of them can get past.
Cool. Question from our Devil Bend group. What part of the waterway is the fishway situated in, and is there water available at all times, or are they perhaps dry at low water times?
So is that particularly the Dights Falls example?
Yeah. Or
Yeah.
just I guess in general terms, the design of fishways.
Yeah, so it really depends on the system. Most of the ones I've been involved with, they have water flowing through them all the time. So then even when you get into, for example, during the summer when it flows down on that one fishway in the Barwon River, you might have very, very little water going over top of the dam wall, and most of the water going around that dam is actually going through the fishway. And Dights Falls, it's that fishway stays wet and flowing year round.
Cool. We do have a question about litter accumulation. So is there much litter accumulation that you've seen in some of these fishways, and that can reduce their effectiveness? And,
yes.
Yep. And does there need to be maintenance to kind of ensure that stays efficient?
Yeah. Another good question, and that absolutely happens. And that happens both in the vertical slot fishways and the rock ramp fishways. So you can imagine, there's litter, there's all sorts of woody debris. So at the top end of the vertical slot fishways, they have basically the metal grills to catch that material, and that does have to be removed. But even in the rock ramp fishways, at the upstream ridge of rocks, you can imagine that after a high flow, say in spring, and you'll have tree limbs flowing down, that accumulates there as well. So anytime after a large flow event, you have to go and clean out that debris, because otherwise the fish can't get around it. It just becomes a new type of barrier if it's not removed.
Excellent. Do you know of any populations that have become extinct upstream of barriers before those fish ladders were installed?
I'm trying to think. Well, really, even in the Yarra River, before the installation of Dights Falls, I think there were only 2 captures of tupong. So although they didn't go extinct as a species, they were extirpated from the Yarra River. And those 2 fish that were observed in the last 100 years before the fishway was installed, I think those were just introductions where someone actually moved them up by hand. I guess a good example of an extinction event would be the New Zealand grayling, and whether and barriers would be one of the potential impacts, but that species did go extinct.
Cool. And just a couple last ones. Are there trout in these streams, and do you know what impact they have on population numbers of the other native fish?
Yep, there are trout. Yep, and no, I don't know the impact. Some of the impacts of trout that have been studied by some colleagues have been in smaller streams and focusing on different species of galaxids, and most notably the ones that don't migrate. So really they've evolved in some of these really small streams, and then they persist in very few streams. And once trout get up into those populations, those fish can disappear. The one good thing about the Diadromous species is even if they're lost from a system, they can recolonize a river very easily because there's a new supply every year, really. So, and given that they don't home to a particular river, there's always the potential for recolonization. So it makes it a little bit easier to manage the Diadromous ones that are migrating upstream.
Excellent. And we just have one last question that's they're asking about what species of the tiny fish that you find in Blackburn Lake are. So I know that I've seen plenty of the introduced gambusia, so the mosquito fish in Blackburn Lake. So I don't know if you've got much knowledge of that area, Frank, but I have seen that Sue from the Blackburn Lake Sanctuary has jumped into this this talk, so she might be able to chime in into the chat as well.
Yeah, that'd be great, because I don't know personally. But you're right, they're probably gambusia, and if they're swimming really close to the surface, it's a good chance that they are gambusia.
Excellent. And that's got to the end of the question. So yeah, thank you, Frank.
Okay, great. So we've discussed the barriers as a potential threat. The next one that we briefly touched on was modified flow regimes. And so, as I mentioned, you can have dams in rivers that hold back flow when we get lots of rain, so you have less water flowing out of the rivers. And with that culvert example, we had some observations that more fish were swimming upstream during increased flow rates. So we really wanted to have a good look at how modified flow regimes can impact fish populations. So we really focused on the downstream spawning migrations, the attraction and immigration into rivers, and upstream dispersal. So those three processes we've had a look at for various species. So we'll start with downstream spawning migrations. And this work was done in, there's some in the Thompson River and some in the Bunyip River, and it involved Australian grayling. They're a threatened species. So we were looking at the movement of adult fish, and we used acoustic telemetry to track their movements. So what we had to do is we had these really small transmitters, and they have a diameter, of about 5 millimeters, so that they're quite small, and they give off sounds, and those sounds can be picked up on receivers. So here on this photograph on the right is a receiver it's got a chain weight on the bottom, a float on the top to keep it upright in the river. And when a fish swims past one of these receivers, it'll record the unique tag number, well, a fish that's tagged anyway, a unique tag number of the transmitter and the date and time. So using this technique, you can track the movement of fish through a river. Then the other thing that we did in association with this, we set some of these really fine mesh drift nets. So this photograph on the left, we set those in the river, and that's to catch eggs or larvae of fish once they'd spawned. And this photograph on the bottom right, that's actually an Australian grayling egg, and they've got a diameter of about 1 millimeter, so they're quite small. And you can imagine that when we set these drift nets, we do catch quite a bit of debris. So we put the debris in these sorting trays, and then row by row, we look at them through a dissecting microscope to pull out larvae and eggs. So that way, this project looked at the movement of adult fish, and then whether they were spawning or not. And I'll quickly go into some movement results. Now, like I say, we did this in the Thompson River and the Bunyip. The Thompson River over 2 years and several years in the Bunyip River. I'm just going to show you a movement graph for one individual, but they were all pretty similar. So this graph that's showing now, the axis here on the left that's marked distance, that's distance upstream. So you can see this one marked here, this dot where it says tagging location. This fish was tagged over 140 kilometers upstream. Now, all these other black dots indicate when they were picked up on a receiver. And this solid line shows the flow, the volume of water flowing down the river. And the scale here is on the right-hand side, and the bottom is just the date. So, all but one of the fish in the Thompson River that were tagged, they all moved downstream to spawning sites during these flow peaks. And the thing about the Thompson River is these flow peaks were created using environmental flow releases. So this one was an environmental flow release, as was this one. The other year where we did the movement work on an Australian grayling, there was only one, but all but one of the fish moved down on these environmental flow peaks. If you notice over here on the left, there was quite a large flow, but you wouldn't expect any movement there yet, simply because it's too early. The spawning period hadn't started yet. Similar results in the Bunyip River. One contrast for the Bunyip River, though, is that there's quite a bit more variability in flow in the Bunyip River. And the people who did that work, they actually observed some fish. They'd move partway downstream on one flow, and then they'd hang out in that area until the next flow pulse came down and moved the rest of the way. So we tested these movements using statistical methods, and yeah, so it demonstrated that these environmental flow releases could trigger the downstream spawning migrations of Australian grayling. The other thing we did, as I mentioned, is we did the netting, the drift netting for eggs and larvae. And what we found is when these fish moved down to these areas to spawn, we actually collected eggs or larvae, so it showed that they did spawn. And then there were 2 years in the Thompson River. This was after we completed the movement work, where we continued to do the netting, but not the movement, where they didn't release any environmental flows for various reasons, and there wasn't any natural rain events that caused peaks in flow in these 2 years, and we didn't catch a single egg in those 2 years. In contrast, whenever there was an environmental flow release down the Thompson River, we would catch hundreds of eggs. So it sort of builds the evidence that if they don't migrate downstream to spawn on these spawning events, or these flow events, they don't spawn at all. And then there's also some work done back in the 80s and this was during the drought period, so there were no flow pulses, and they looked at some female fish in the upper reaches that hadn't migrated down to spawn, so they were still residing in these upper reaches, and they had started to resorb their eggs already. So that's an indication that they weren't going to spawn at all. So it really builds the picture. If they don't have these flow pulses, they won't migrate downstream to spawn. So then the next process we were going to talk about was that immigration, or the attraction into river. And we did this in several rivers, as far west as the Barwon River, so around Geelong, and as far east as the Tarwin River. So we picked 5 rivers, and we did this over 3 years. And these rivers, they all have sort of the different catchment sizes, so quite a bit of variability between the rivers and the amount of water flowing out of them. And there's also variability in the amount of flow between years. So it gave us a large range of spring flows. And like I say, we're trying to determine whether the volume of water flowing out in the ocean impacts the number of these fish immigrating into rivers. So we did all this sampling in the lower freshwater reaches. And once again, this work involved catching fish using nets. So if you look at this photograph on the top, on the right, that's actually our net in the Tarwin River. So it's set so it's catching fish moving upstream. So if fish or several fish are moving along this bank, they'll get caught in the net, and actually the body of the net's quite large, so it can hold can hold lots of fish. In the Barwon River, we did it using the fishway trap that I showed you previously. And like all our projects, once we catch our fish, we have to identify them, count them, and then release them at the site of capture, or in this case, upstream of the site of capture. And just to give you an idea, this photograph on the bottom right, that's a 20-liter bucket, so it's less than half full, and you actually see some of the small galaxids up on the trying to jump out of the water. But this is about 9,000 fish, so it just gives you an idea how small these fish are. And we would catch anywhere from less than 10 fish in a 24-hour period. I should add, this sampling was done for this project over a 24-hour period. But our highest catch, we caught over 86,000 fish. Now, of course, we didn't count each individual fish. We did subsamples of counts, and we used the weight of those fish and applied it to the weight of the entire catch to determine how many of each species, or to estimate how many of each species. So like I say, we did this over 3 years. Now, these nets were set through a good portion of the migration period of these species. So we would start in late September or early October, and that would go right through to early December. And these nets were set anywhere from once per week, and sometimes it was twice per week. And then we'd go and catch these fish at each of these sites at least once per week. So a fair bit of effort. And so what we found that more fish immigrated into rivers when you had more water flowing out in the month of September. So this graph, it's really some of the outputs from our statistical test, but this axis on the left is the probability of having a high catch rate in any one of our sampling periods. So those 24-hour sampling events. And this bottom axis here, that's just the mean discharge during the month of September. So how many megaliters per day was flowing down the river. And the probability of having a high catch was significantly higher when you had higher flows in your river. So this demonstrates that when you have more water flowing into the ocean, you get more fish moving into and up into the freshwater reaches of these rivers. So that hadn't been demonstrated before. I guess perhaps the downside of this, you can imagine at the top end on the right side of this graph, 2,500 megaliters per day for a month. That's a lot of water. So we can't really use a technique like environmental flow releases to try to attract huge numbers of fish into rivers. So we really need to rely on some really wet springs once in a while to attract a lot of fish. But environmental flow releases might be able to have an impact at the left-hand side of this graph, in the really dry rivers. So if you have a very dry year at a dry river, you may be able to use an environmental flow to attract some fish in just to maintain some populations in those rivers. So that's what we found out quite recently around attraction into rivers. And then the third process we were going to talk about today was that upstream dispersal. And this builds on some of those observations we had in Cardinia Creek when we observed some more fish moving when there were these slight increases in flow. So this was our attempt to try to show whether that actually makes a difference or not. So this work was completed in the Glenelg River and the Moorabool River. Both of these are west of Melbourne. And similar to some of the other studies that we've done, we had control sites. So in the Stokes River, which is a tributary of the Glenelg River, these white dots are our sites. And then in the Barwon River. So at those sites, there were no increases in summer flow pulses, whereas in the Moorabool River and the Glenelg River, they put down what they call summer freshes. So it's a fairly short period of relatively small increases in river flow or discharge. And so the total duration is about 10 days. So that includes the period where it's increasing to the peak. The peak might only last 3 days, and then it starts to decrease. So that they're not really large flow events. So once again, we used those double wing pike nets catching fish moving upstream. And our expectation was, if this makes a difference, then our sampling during the flow releases should catch more fish moving upstream than during base flow periods when these flow releases aren't occurring. And so this slide shows one site in the Moorabool River, and the top image is during the base flow conditions, and the bottom image is the same site during the environmental flow release. So you can see that both water velocity and water depth are greater during the e-flow release. So it does make a difference in the river. Then the question is, do the fish detect this? And given that there's this cue, do they follow it? Do they say, okay, there's more water, let's migrate upstream? And so once again, we did netting before the flow release and after, or sorry, before and during at our impact sites and at control sites. And we did this, we got results, for 3 species, short-finned eels in the Moorabool River, and then common galaxias and tupong in the Glenelg River. And in all cases, our catches were higher during the flow releases than they were prior to. And the numbers I'm showing here, that's relative to what happened at the control site. So for these really small eels, catch rates went up quite substantially, 26 times higher. For common galaxids, it was 6 times higher. And for tupong, the effect wasn't quite as large, but they were about 40% higher, so 1.4 times. So this demonstrates that upstream movement significantly increases if they have these flow pulses going down the rivers. The one thing I'll add before we get away from this part of the flows is that you can also have an interaction in these threats. So flows and barriers can impact on any sort of management technique that you're using. So for example, you have to be careful about trying to use an environmental flow release to get fish to disperse upstream if there's also a barrier that'll stop them from moving. And I know it seems obvious, but it's something that we always have to be careful about whenever we're coming up with some management techniques to get fish to improve fish populations. You have to make sure that it can work given the condition of a particular water body. So that really talks about how flows impact those 3 processes. But similar to the barriers, there's a question about, so does that impact populations at that much larger scale? So we've done a bit of that in terms of flows as well. And this work was done through long-term monitoring, where we do electrofishing in the rivers to look at population size and the size structure of fish in the population. It's been going on in the Glenelg River for years and in the Thompson River as well. So what we looked into here was a change in environmental flow releases in the Glenelg River. So prior to the summer of 2017, base flows could be quite low in the Glenelg River. And they could get so low that during some dry periods, you'd really get just these isolated pools with maybe a trickle of water going between them. And you can imagine that really doesn't facilitate this upstream movement of fish at all. So starting in the summer of 2017, they increased the base flow, so you had more water flowing down the lower reaches of the Glenelg River, plus those fresh releases that we spoke about in that previous section, 1 to 2 a year in either summer or during really early autumn. So that's the change. And we wanted to look at whether the fish populations changed as a result of the change in those flow releases. So we used the Thompson River as our control river in this case, and throughout sampling there, base flows had been high enough to maintain some really decent flow during the summer to allow that upstream movement almost continuously. So that's the approach we took here. This just shows you a map of the Glenelg River. So all our sites were in the lower couple reaches of the Glenelg River, and these were monitored annually. So we would go in early autumn and we sample at these sites, and that's been occurring for quite some time. And so once again, we count, identify, and measure the fish that we capture, and then we release them back at the site of capture. And so what have we found? These graphs, once again, the axis on the left shows our index of abundance, the catch per unit effort. The top graph is for tupong, and the bottom graph are for common galaxids, two species that we catch in the Glenelg River, Diadromous species that is. And once again, the vertical dashed line indicates when that management implementation occurred. So as you can see, the catch prior to the increase in the base flows was typically quite low for both species, and significantly lower than what we found in the past 4 years. And once again, our statistical testing shows that this is statistically significant, and it contrasts with the Thompson River, where the catch rates were quite similar between these two periods. So that really shows that at a population level, you can impact fish, using flows or environmental flow releases as a management technique. So when I guess into the future, one of the things that would be nice to do, it'd be great to look at not only the Thompson, the Glenelg rivers, but add a number of rivers, so we can start to look at a broader picture of how flow impacts these populations. So autumn and winter flows that could drive spawning, spring flows that can drive the attraction and immigration, and summer and early autumn flows that can impact the upstream dispersal. Are there trigger points where having it flows too low at one of these times, will that limit a population completely? So that'll take a lot more sampling and in a lot more rivers, but it might in the future give us a better way of deciding when and where to use environmental flow releases. So finally, I'll finish off and then we can go back to questions, because I'd just like to thank the large number of people that have been involved in planning these research studies and collecting the data. As you've seen, it's quite a lot of work. It's been going on for a number of years, and we have a small army of people who have helped along the way. And I'd also like to acknowledge Melbourne Water and the Victorian government for funding. So Melbourne Water funded the work that was done in association with barriers, and the work that was done in the Bunyip River, and DELP through the Environmental Flows Monitoring and Assessment Program funded most of the work related to the flows studies. And at that, I'd like to say thank you. I appreciate your time, and we can now probably take any additional questions if there are any.
Excellent. Thanks, Frank. Yep, we do have a few more questions that have come through. So we have a question around environmental flows. Does storing water affect the temperature of the water when it is eventually released? And does it have a flow-on effect on the fish populations in any way when you release
it? Yes, it can. So it'll depend on where the water comes from out of the reservoir. So if it's released from the lower parts of the reservoir, so if the opening's deep water, that water can be colder, and that can impact some fish species. So species, for example, like blackfish, you could end up with cold water that could impact their spawning.
Cool. We've got a couple of questions about chemical attractions for fish. So is there any work on the effects of the catchment vegetation that could create a bit of an attraction of a stream, i.e. like a habitat indicator rather than conspecifics? And yeah, related to that, so along with if there's vegetation kind of chemical attractants you could create for fish, could you create similar olfactory cues used by fish to artificially attract them to waterways, so creating a bit of a fish perfume maybe?
Yep. So the answer to the first question is we don't know whether there could be whether plants could be driving that chemical change or the chemical attraction. We just don't know. And the second question, if we could determine what the fish were picking up on, then yes, there's no reason you couldn't try to attract them in using those particular chemicals.
Excellent.
It'd be an interesting thing to try. But I will add, there's also some work being done on vegetation and the impacts of flows on vegetation. I'm just not particularly familiar with the results.
Cool. Apart from the impact from barriers and flows on fish population, could the fish population also be affected by the presence of any environmental pollutants?
Yes. Yep. Yeah. I've just touched on these two threats. They're two of the big ones for diadromous fish. But pollutants, absolutely. Non-native species. There are all sorts of other negative impacts that can happen with our native fish species.
Cool. We got a question about the diets for fish ladder. Are you aware of the redesign of it? Were you involved in that? And do you know what might have contributed to making that redesign?
Yeah, I wasn't involved with the redesign. I am familiar with it. So what occurred at the Dights Falls Fishway when the trapping was occurring, and it was showing that fish that can get into that entrance cell or that pool can make it all the way to the top. What they found was when flows were a bit higher, and I think it was maybe around 1,000 megaliters a day, they were always catching fewer fish getting into the fishway. And then upon review, you could actually people could see it, but they did some hydrological modeling anyway. But what happened was the entrances to the fishway during those higher flows, there was a huge amount of turbulence, so it looked like it was really impacting on the fish being able to get to those openings. So that's what sort of determined that there was a problem there. And my understanding is that the work was to create another opening that is farther away from the weir wall. So when water levels up, then that new opening will be flooded, and it'll be sheltered from all that turbulence. So that should allow fish to be able to find that entrance, which they couldn't do before. And then I think what will happen is there'll be some sampling around that to determine whether it's been effective or not.
Thanks. How can you be certain these particular interventions are leading to overall fish population increases and not seasonal factors, for example, floods?
In terms of I'm trying to think, is this more the flow stuff, I assume, in the Glenelg River?
Possibly, or with the fish ladders. I guess it might be a question around can you split out some of those larger factors?
Yep. So it's a good point. So in terms of the barriers, so Dights Falls, for example, if you get a really big flow pulse, and I can't remember, it might be between 13,000 and 16,000 megaliters a day, that will actually drown out that barrier. So if that occurs, fish can get around it, or they could just swim right over top of it. So that's a way some fish would get past that barrier. When you look at the hydrograph of the Yarra River, that's a fairly rare event. And when it does occur, it usually occurs in winter outside the migration period. So if that occurs, for example, in July, there's no reason that some of the fish from downstream couldn't get past that barrier. But what'll happen is it'll happen in much smaller numbers, because all that higher mortality that we were talking about, because you have that really high concentration of fish, that would have already occurred. So it can get fish past barriers, but it typically doesn't happen during that migration period in the Yarra River.
Thanks. Do you know how much it costs to build a simple fish ladder? So we've got a question about like a barrier of about 2 meters, for example.
No, I don't. So a 2 meter barrier, it's got to be in the millions of dollars.
Cool. Yeah, and these projects do tend to be fairly costly.
They are, and yeah.
Does silt load affect fish numbers, for example, with increased development along waterways and the increasing, I guess, turbidity and sediment loads?
Yes, and yep, it certainly can. And I'll go back to blackfish as a good example of that, because they spawn in hollowed out logs, and so if you have a lot of sedimentation that covers those eggs, they end up dying just because they don't get any more oxygen.
Excellent. And I think we've hit the end of our questions there. So I might hand back over to Teresa now.
Thanks, Frank. That was a great presentation. So just before everybody heads off today, I just want to give you a reminder that on Thursday, the 3rd of September, we've got investing in Western Waterways. Then on Thursday, the 17th of September, we've got strategic pest animal management. So the recordings will be available on the website, and you'll also be notified by email so when that comes up, so please just be patient with us. And if you do have any questions, please contact us on waterwatch@melbournewater.com.au, so the usual Waterwatch email address. And I just want to say thank you again to Frank. Thanks again to Rich and also Elisa and all of you for showing your support and coming along again. So thank you, and yeah, stay safe and see you in 2 weeks' time.